This is Grace, Owen, Dylan, Julia, Kaylee, and Jessica from the biology department at Northwestern College. We annotated 2 phage genomes this semester, Tedro and BAjuniper, both of which were discovered on the host microbacterium foliorum.
Tedro was discovered by direct isolation on M. foliorum by Garrett Raymond and Erica McKinney using soil collected in Hayward, Iowa. Amy Holstein discovered BAjuniper by direct isolation on M. foliorum from soil collected in Orange City, Iowa. We purified the phage through at least 3 rounds of purification. Tedro produces plaques that are round and clear and average 1 millimeter in diameter. BAjuniper's plaques have an irregular morphology and are larger, with a mean diameter of 6.1 millimeters. BAjuniper's plaque size is less uniform and more turbid than Tedro's plaques. After we prepared DNA from our phage high titer lysate, we sent our samples to the University of Pittsburgh for sequencing. Tedro was assigned to cluster EF based on genetic similarity and BAjuniper to cluster EB. Both clusters consist of lytic phages. Tedro's genome is just over 56,000 base pairs long and is circularly permuted. BAjuniper's genome is almost 42,000 base pairs long, and it has 3' sticky overhanging ends that are 10 base pairs long with the sequence ACTCCC GGCA. Both genomes have a high GC content.
Tedra's genome shows quite a bit of similarity to the genomes of other cluster EF phages. You can see that by the purple color when we compare Tedro to anakin and quantum. BAjuniper's genome is less similar to other phages in its cluster EB. Here we are comparing BAjuniper to avocado man and skyboard, where our phage is similar in some regions and quite different in other regions. When BAjuniper's genome was auto-annotated, it included many genes included by white rectangles that were not actually genes. We saw this happen with one of the phages we annotated last year too. This reinforced for us the importance of human annotation. Tedro and most cluster EF phages have 2 genes identified by bioinformatics as DNA E, like DNA polymerase III alpha. The 2 genes differ dramatically in size, with the first gene approximately half the size of the second.
We were curious about these 2 genes, both called DNA E like DNA polymerase III alpha, but so different in size. We dug into the literature and found that DNA polymerase III has 3 parts. It is responsible for the replication of bacterial genomes, and it consists of DNA polymerase alpha subunit, processivity factor beta clamp, and clamp floater complex. There are 2 forms of DNA polymerase III, DNA E in E. coli and others, and Pol C in low GC content bacteria like B. subtilis. The polymerase or alpha subunit of both Pol C and DNA E has a similar structure, including palm, thumb, and finger domains with a PHP domain, oligobinding domain, and a helix hairpin helix domain. M. tuberculosis has 2 DNA E genes. The second is called DNA E2 and is believed to be involved in error-prone translation DNA synthesis.
Our professor learned about an AI-driven structure prediction software called AlphaFold at a C faculty meeting. We compared structures for Tedro 56 and Tedro 57 to structures for DNA3 alpha subunits from various organisms by running sequences through AlphaFold and looking at the HH spread analysis. Tedro 56's predicted 3D structure is similar to the crystal structure of the first half of DNA polymerase from M. tuberculosis, and even more similar to the predicted structure for DNA E-like DNA polymerase III alpha from Clostridium perfringens. Tedro 57's predicted 3D structure is similar to the crystal structure of DNA polymerase from M. tuberculosis. HH spread analysis for Tedro 56 suggests that it has domains that resemble PHP and polymerase from histidinyl phosphates, that domains with 100% probability and 93% coverage. HH spread analysis for Tedro 57 suggests that it has domains that resemble nucleotide transferase, PHP, NTPase, alpha subunit finger, helix hairpin helix, and oligonucleotide binding domains with 100% probability and more than 98% coverage.
We wonder if Tedro 57 may be a DNA E-like DNA polymerase III alpha. Tedro 57 is 31% identical to DNA E-like DNA polymerase III alpha subunit from M. tuberculosis, with 50% similarity. Tedro 56 may be a truncated version of this gene that could function in DNA repair, like DNA E2, but structural similarity to DNA E from C. perfringens. These two proteins are 30% identical and show 47% amino acid similarity. We have some work to complete before the end of the semester. We're finishing up our annotation of BAjuniper and are writing a microbiology resource announcement. We have many people to thank for making this research possible. The C Pages program, HHMI, the Hatfield Lab at the University of Pittsburgh, especially Dan Russell and Northwestern College, especially the Department of Biology.